Hello everyone, I decided to do my journal article on one titled Environmental Nuclear Magnetic Spectroscopy, an Overview and a Primer by Andre and Myrna J. Simpson and Ronald Soong. First, I'd like to talk more about what NMR is exactly. You guys are probably familiar with it by now since we've been doing it for, the past 2 semesters or something, but I will explain it anyways. It basically stands for Nuclear Magnetic Resonance Spectroscopy, and it's an analytical technique that is used to determine the molecular structure of matter in many states. These states can include solids, solutions, or gels, just to name a few. NMR is also described as a non-selective detector, which basically means that it can detect any molecule that contains an NMR active nucleus, but the most common atoms used are hydrogen-1 and carbon-13. NMR can also range from being one-dimensional or multi-dimensional experiments, and an example of a multi-dimensional experiment is to identify bond connectivities or spin interactions. Two-dimensional NMR experiments can help to identify unknown compounds that are present. This is pretty similar to mass spectrometry. Now, the journal also included a table with commonly used NMR techniques. The ones on the left are the most used in the chemistry book. So the first one is solution state NMR, which is the most common and the one that we're most familiar with. It provides the highest resolution, and it can only be used with soluble components, and the approach is also fully quantitative. Solid state NMR is important for environmental analyses because it gives insight to the types of carbon in a sample. And thirdly, the high resolution magic angle spinning NMR, also called gel phase NMR, allows for swollen components to be studied with a resolution similar to solution state NMR. Now, the journal also included the most commonly used atoms in NMR spectroscopy, and we're mostly familiar with the hydrogen-1 and carbon-13, but there's many more. Like lithium, nitrogen, fluorine, aluminium. Now, like I mentioned previously, you can detect pretty much any atom as long as you have an active nucleus in the NMR spectroscopy. So now that we know more about what NMR is exactly, we can go into why or how it's used in environmental research. Basically, ecosystems are constantly changing due to many factors such as climate change, urbanization, agriculture, and industry, just to name a few. There's many. And it's important to understand the processes involved in keeping these ecosystems sustained. And with that information, we can help to prevent, combat, and mitigate these changes, as well as reverse some of the negative side effects that we've caused. And some of the applications of NMR that have been used in the past in environmental research include developing biofuels, treatment for pathogens, investigating chemical ecology, and detecting pollutants. In the journal I was researching looked at 3 specific categories of environmental concern, specifically structure, which talks about the structure of soil, air, and water, interactions, which discusses the interactions between molecules and the impacts that they have, basically saying how living systems respond to changes in their environments. So for the first category, which is structure, I think it's important to talk about the first application of NMR in the environmental science field, and this was to analyze soil organic matter, which is abbreviated as SOM. Now, SOM is able to bind with metals, organic pollutants, and minerals, and therefore it's important to know the structure of soil to understand its interactions with these materials. SOM stores more carbon than the atmosphere, which is really interesting to know, fun fact. And the NMR is useful to figure out where carbon goes when it leaves the atmospheres. And cross-polarization, magic angle spinning, solid-state carbon-13 NMR is the most common technique used to study SOM because it's easy to prepare samples with soil, as opposed to using solution-state NMR. NMR can also be used to identify pollutants and their corresponding degradation products. An important experiment that was conducted in 1976 by Taves et al. Used fluorine-19 NMR to discover perfluorinated organic fluorine in human plasma, and to do this they used liquid chromatography and NMR to identify these compounds. The second category talks about interactions, and going back to soil chemistry, to properly investigate SOM, it's important to make sure that all components of the soil are examined, specifically the pore water, which is essential for nutrients to transfer to the roots, and the soil-water interface, which is basically a measure of the rate that contaminants can travel between water and soil. It's also important to examine the soil's physical and chemical properties under different hydration levels. There are two types of ways to study environmental interactions. The first one is one that detects free and bound species, and this basically means that chemicals bind to many different components and to varying extents. This is useful, but the data can be complex and difficult to interpret because all of the signals appear on the same spectrum. The second method is a more technical approach because it only targets bound species, and this makes the spectrum easier to interpret because only the bound species are present. That may have been hard to understand just by words, so the journal had this diagram that's super helpful to use. Basically, the top image, the top spectrum, showcases two different types of components, which are fluoronaphthol and PFOA. And it's hard to detect either one of those components because they're all kind of mashed together. But when you look at them isolated, which are the middle, left, and right spectra, it becomes easier to see their specific components and their structures. The third category talks about impact, and the journal specifically discusses metabolomics in this section. Metabolomics is the study of small molecules that are found in cells, biofluids, tissues, or organisms, and these molecules are called metabolites. And metabolites are affected by many changes to their environments, specifically pollutant exposure, nutrients, temperature, pH, and salinity. NMR is used on metabolites that are significantly stressed by changes in their environments. Now, this field of study is very significant because with it we can learn more about how changes to our environments can affect our ecosystems, and using NMR has proved to be very helpful because with it we can detect early warning signs that we may not have realized otherwise. Before I read this journal, I didn't really realize how many uses NMR spectroscopy had, but after reading it I learned that you can study the structures of soil, air, and water to gain more knowledge about their chemical makeups, specifically whether or not they have pollutants in them, and what we can do about it. We can also assess soil's physical and chemical interactions when exposed to xenobiotic chemicals like pathogens, drugs, or contaminants. We can also infer on how ecosystems are affected by changes to their abiotic environments. And all of these help us to come up with ways to protect our ecosystems and suggest solutions to the many pressing environmental problems that we face today. Anyways, I hope you enjoyed what I talked about today, and if you want to check out the journal, I will leave the title in my comment and link it there. And I hope you have a good day and good luck on finals, everyone. Bye!